Thank you, AWS, for the invitation to be here today. It's our pleasure, Abhishek and mine, to represent AbbVie and tell you a bit about how we're using some of the biological foundational models to drive small molecule drug discovery. So we just heard a lot about the large molecule side, so we're going to go deep into the small molecule side. All right, I wanted to take just a minute to tell you a bit about AbbVie, if AbbVie is a name that maybe you haven't heard. We are a rather new organization. So we were spun out of Abbott in January of 2013, so we're only 12 years old. However, we are not a small company. We have state-of-the-art R&D sites as well as manufacturing sites across over 20 countries. We employ over 50,000 employees in over 70 countries. And I will say that our mission is really to find innovative medicines to treat human disease. And every employee that I meet at AbbVie is absolutely driven by that North Star of delivering these innovative medicines for patients. And it's not just me saying that. AbbVie is a company that year over year has won the accolades of best places to work. So it's not just me, it's really a fantastic company. So in my role, I support research IT, which encompasses not only your traditional IT, but we're also a group that works extremely closely with our scientific stakeholders to deliver capabilities, and that includes data engineering, data science and visualization. It includes machine learning. Abhishek is our machine learning lead for AbbVie research. It includes high-performance compute. It includes software engineering. So we have a team that's really there to support all of the R&D scientists. So what does R&D work on at AbbVie? There's some therapeutic areas that are shown there. Our main three pillars are oncology, immunology, and neuroscience. But we also have a footprint in eye care, as well as in medical aesthetics. And we do have some specialty, mainly they're indications that we've brought in through mergers and acquisitions. These can include migraine, pain, obesity, as well as a few others. But it's not really just about the therapeutic areas. Within AbbVie R&D, there's also these 7 innovation pillars, if you will. And the way you can think about those are true horizontals of innovation that are really agnostic to a given therapeutic area. So if you think of things like genomics or precision medicine in your specific company, right? Those are there to enable finding those right targets, ensuring that you have the right biomarkers for your clinical studies, ensuring that you have identified the right patients in those clinical studies. So it's really that entire end-to-end pipeline where these types of capabilities come to life. Therapeutic modalities, right? We do a lot of innovation around therapeutic modalities. We heard about large molecules. Everybody's very well aware of small molecules. But we do a lot of research in genetic medicine. We do a lot of research in intravitreal injection for eye care. We do a lot of antibody drug conjugates. How can we actually bring combination therapies together? We do work in dual variable domain antibodies, degradamers, molecular glues. The complexity of modalities and how we deliver medications is becoming more and more complex every day. So the innovation, we're there to really assist our researchers and, of course, use machine learning and AI approaches in any of these spaces. In fact, AI is called out as one of the pillars of innovation, and it's really just something that's embedded in the work that we do every day. So AbbVie is really kind of getting very excited about AI. And I was thinking about, like, why is it that AbbVie, as well as other companies right now, it's like everybody's talking about AI and agentic AI, but AI nonetheless. It's really what I see as coming together of this trifecta, where AI sits right in the middle, and it's like all 3 pieces really have to come together. And I've been doing this for a little while and kind of watching, like, data was really becoming good. And you have a lot of this compute that's becoming good. But you got to really have the data, the compute, as well as the math. That all has to be there. So within AbbVie, data has been a major focus. I would say over the last 5 years, we have built one of the largest knowledge graphs in the industry, which we call AbbVie's R&D Convergence Hub, or the ARCH. where we're taking internal, external data, we're doing all FAIR principles, all the ontologies, taxonomies, terminologies, harmonizing all of that, extracting the knowledge so that we can easily apply machine learning algorithms to that knowledge graph. And we're now just starting to reap the benefits of all of that hard work. Compute. So as I mentioned, I do support high-performance compute at AbbVie. We do have a footprint both on-prem as well as in the cloud, where AWS is one of our preferred partners. And we are now able to meet the needs of all of the machine learning capabilities that our scientists want to be able to use on a regular basis. And last but not least is algorithms. And this has really been a lot of fun, I think, to see, especially in the past couple years, where the open source format of math, right? The ability to share math, and whether those be through repos, whether those be through academic collaborations, whether that be through platforms like Bedrock, where we now have access to use large foundational models that we as a company cannot create ourselves and fine-tune those and do inferencing on those. It really has changed the way that we can approach AI just in our everyday work. So I think that's why at AbbVie it's a really great time to be interested in employing and using machine learning approaches to our daily work. So where are we doing artificial intelligence? Well, at AbbVie, we're actually using it kind of everywhere. So here I just have research development and on market. In research, like many of you who are also in biopharmaceuticals, we use it for target identification, target validation. We use it when we're identifying lead molecules, whether they be small molecule, whether they be large molecule. We use it for optimizing our leads in the research space. In the development space, clinical trials, there's a whole track, right, on clinical trials. And using machine learning or artificial intelligence approaches in the clinical space is essential. Right, there's a lot of efficiency gains that we have seen ourselves, and I know other companies are seeing as well, when you start to drive that efficiency in your clinical trials, getting those medicines to patients faster. And then on market, so it doesn't just end in the research space. Once things are on market, we can actually go and use machine learning approaches to look at some of the like risk indicators, or we could use real-world evidence and predict patient journeys. Things like that become very powerful use cases for machine learning and artificial intelligence approaches. So Abhishek and I are going to go deep into research, and that includes target identification, validation, as well as how do we optimize our leads. And why are we so interested in small molecules? I mean, you know, it's a lot of fun to talk about some of these new complex modalities, but at the end of the day, AbbVie, our core pillars of oncology, immunology, and neuroscience are still driven primarily through small molecules. And small molecules do represent a very patient-friendly mode of drug delivery, and that's very important for adherence, et cetera. And it's also, small molecules are not going to be going away. 60% of FDA-approved molecules are small molecules, and not only as independent therapies, but as combination therapies. I don't think small molecules will be going away when we think about antibody-drug conjugates, combinations, especially in the oncology world, where we're looking at co-dosing and sometimes tri-dosing. All right, at that, I think I'm going to turn it over to you, Abhishek, to start talking about models.
Thanks, Jen. First of all, I'm between you and lunch, so hopefully I'm entertaining enough. So how do we do drug discovery? How do we actually cure patients? Now, whatever I'm going to talk today is all about small molecule, but similar thoughts and process hold true across all modalities. If you ever cure a patient or you want to cure a patient, that's how you would actually go with the process. How we do it is essentially we first try to identify a target. We want to actually understand a target that can be druggable, ligandable, and all the right things actually with that. Now, that's a more difficult thing than actually just said, like, okay, let's find a druggable target. Because if you think about it, a disease is a very complex thing. Example, cancer. That's why we throw a kitchen sink at it. What we do right now is we identify one single target and say we'll cure cancer. It's not that simple, actually. We have to have multiple targets. We are not there yet. I'm not saying that we are there yet. And not there as in like, not just we, but nobody is there yet, actually, frankly. We are in process of going in that direction, but ideally we should have multiple targets, multiple molecules. Now, that's a story for a different time. But the point is that we want to identify a target, one single target, that will alter the cure of disease. I said alter the cure of disease. It sometimes cures, and sometimes it just changes the process, actually. Now, with that being said, actually, what we want to develop is models. So once you actually have target, and I'm assuming this story that you have identified targets, because there are a million ways to come up with new targets. There are like, as the time progresses, there are like more and more ways. There are machine learning ways of doing it. There are assay way of doing it. There are like CRISPR. There's like so many ways of doing it. Cell painting. There are like so many ways of coming up with this new target. Let's assume that you have a disease and you have come up with a bunch of them. Now your problem is among 100 targets, or probably more than that sometimes, you have so much of information because there is literature backing some of them, there's literature backing some of not that is there. How do we actually understand which target to choose? Because you can choose one, just one. So what you need to do is you need to build algorithms, and this is just a perspective. I'm pretty sure somebody else will have a slightly different perspective to this. But what we want to build is algorithms which can predict druggability, which can predict ligandability, which can predict causality, and then probably the mode of medicine down the line in the sense that, do you want to use small molecule? Do you want to use large molecule? Do you want to use something else actually down the line? So with that being said, actually, what we want to do is we want to actually use some of the biological foundation models, actually, that can accelerate the identification, as I said, to identify essentially the candidate, the target, as well as the molecule. You want to actually understand the properties, because once you have identified the target, then you want to build a molecule that have all the right properties. Then you want to actually generate essentially novel structures. Machine learning can help in all of them, actually, frankly. So this is really exciting. You want to optimize the molecule, for sure, right? And then you want to actually also find the hidden patterns, because this is one thing that is also very interesting, because most of the time, actually, when we start a project, many times we do not understand like what's like, for example, mechanism of action, like the true mechanism of action. Of course, there are like multiple hypotheses that you have to test through, but you do not exactly understand some of those things. So finding the hidden pattern, machine learning is very good at finding patterns, and that's what actually we would like to reveal. And then, of course, there's a lot of things that goes on, high throughput screening and all of that, that hopefully can be helped with machine learning. With that being said, this is one example. For example, as I said, you want to identify a target, one single target that will alter the course of disease. How do you do it? Start with algorithm. So this is a paper you can go read, actually. It's druggability. So on all 20,000 human proteome, including like the known ones as well as the dark genome, where you can actually, we have built model, which is highly explainable. So this is another part which is very important. So what most of the machine learning people do is that they build models, and because the users are scientists, and you can't exactly explain most of the time what is going on, the usage of it becomes limited, because scientists are not like believers. If they understand something, they'll use it, actually. So that's why what we have done is like, we took the protein, we featureized it into like multiple different branches, and then we built like small, you can call it, these are not large model at all. You can just probably deploy it on CPUs. You don't even need GPUs, actually, for this probably. But we do need, by the way, but I'm just saying that you can probably just live without it. The point is that we can explain, because we have divided the protein into multiple different features, and we can say which of these features are most important. You can read in the paper, there are some features which are dominating most of the time, because we have more information, the number of features for them are a lot more than the other types. But nonetheless, and it does include some of the chemistry features as well, like, for example, protein, essentially the pocket volume and stuff like that, that is included. And what we found out is that we tested it out on particular target that was known as dark genome, actually. And then eventually there was evidence that people found that it is a viable target and people should target it. So essentially, this druggability paper was, of course, validated in that way, but there were multiple other internal experiments we did actually on druggability. So once you have found out that your target is druggable and at to what level, because there are hundreds of them, then you'll have multiple scores that you'll get actually. Then ideally what you would like to do is essentially ligandability, because something is druggable, and as I said, essentially we have done it on a bunch of like proteins, 20,000 of them, so all of the human proteome actually that we can get our hands on. But once you have the druggability is not everything, because druggability happens at the very end. If this particular molecules make into a drug, it's a very tall ask actually. Before that, you should actually even understand, is it ligandable or not? So in ligandability, actually, there are two ways we can do this. We have chosen to do it in a more non-traditional way, which is using embeddings. But essentially what we did was either you take the protein. So right now, you just have target. There's nothing else, actually, no other information. So you take the target, and then you essentially handcraft the features the way we did earlier, and then you can actually try to predict something in terms of ligandability. Now, the other way around is essentially take ESM2 kind of large foundational model, generate embeddings for your proteins, and that essentially can be used for prediction. And what we found out is that that's a very reasonable hypothesis, actually. Of course, we haven't published anything on this, so I don't have any results in general. But what I'm saying is that this is a very reasonable hypothesis that essentially will hold true, that you take a protein, your target, generate embeddings out of it, and then you can correlate it to something that you want. Now, once we have been talking about this target, let's assume that we have, and as I said, we still are in process of making like causality and mode of medicine, those algorithms will still follow. We haven't built that yet. But eventually, once we'll have all slew of them, hopefully you'll have like, this is druggable, this is ligandable, this is causal with the right amount, and then you can start probably a program. Now the problem is that how do you then identify a molecule? Now, there are multiple high throughput screening methods of doing that. But then there are some machine learning methods that are coming up as well. And this is one example that I'm just showing. So what you are doing in high throughput screening is that you are trying to essentially identify a molecule that binds to the target. So you have one target. You actually make a full three-dimensional map of it using AlphaFold or using cryo-EM or X-ray crystallography. So essentially, you can think it like a big Lego block with bunch of places where you can fit small Lego pieces, actually. And then you have a molecule that essentially should fit in that particular Lego block piece. Now, how do you do that? There are so what I said, by the way, just to explain to you guys, this is a very simplistic version of it. Actual binding does not happen that way. It's a very physics-based phenomena, and there is a lot of things actually that go beyond it. But the point is that essentially when two things bind, the combined energy of them is smaller than the individual energy summed together, actually. So anyway, so once to identify a binding molecule, whichever molecule binds, that could be a program starter in many ways. Now, there are multiple ways to do this. One is essentially, which is traditionally being carried out a lot, where you have a target and you have a lot of molecule you have information on that particular target. So essentially, you just use the molecule and build almost in the traditional manner, it's called many times as QSAR as well, but essentially just build a model for that particular target. So target-specific model, I won't say it exactly QSAR because you can actually build graph-based model. So you can go like really high-end in terms of machine learning in that, and you can essentially predict for that particular target the binding affinity for that particular target. And you can use, as I said, either graph-based method or you can use language-based method. It does not matter as long as you are using one of those methods that you are actually featureizing your molecule on. That's what I'm writing here. I don't have a pointer, but essentially featureization is an important piece. So when you featureize your molecule into a language model, essentially it's a smile string. When you are featureizing your molecule into graphs where each atom is a node and edge is a bond, actually. So anyway, there are multiple other featureization that can exist as well, by the way. This is not end of it, but these are at least two featureizations which are very, very popular in general. So with that being said, actually, there's another thought process that can be brought to this picture. Now, When you actually identify a target, there is no information. You don't have any information, actually. Then what do you do, actually? So then you have your target and your molecule coming together. Now, one machine learning way of looking at it is that you have a protein language model, which is featurizing your protein. You have a graph-based model, which is for featurizing your molecule. And then you are putting them together to predict something. Now, unfortunately, this process is more difficult than just this. But what I'm saying is that that's one method that people are publishing a lot. And that's one thing that you would see a lot of publications, even in, like, I'm not talking about earlier. Right now, actually, those publications are happening. So with that being said, as I said, that one idea would be something like this, where you put essentially your ligand or your molecule, you put your protein is featurized by something like ESM2 or something like that. Your molecule is featurized by some version of GNNs. And then you bring them together by a cross-attention. So there are two types of attention, if you guys are not aware. There's self-attention and cross-attention. And attention is all you need is the paper that you guys should be reading if you haven't read. That's what led to all of this machine learning, ChatGPT glamorization that has happened. But nonetheless, the point is that what you can do is a cross-attention between the protein embeddings and the molecule embeddings. And then you can try to build a model. Frankly, if you ask me, this is just one step. There are multiple steps that you have to do actually to make it work. We are not there yet, but I'm just saying that this is one idea that you can use. With that being said, actually, AWS is a very important piece here. For example, the ESM2 embeddings generation. You need to be deployed somewhere to scale it up, to actually generate those embeddings at scale. This is a very important piece, actually, for drug discovery. Because if you are struggling through these basic things, I don't know when you'll get to the actual science of it, right? So with that being said, AWS has been very helpful in general in our usage of doing things. This is just basic pipeline for deployment that we are using. If you see, there's no AutoML actually used here. But this is just the basic version of how you deploy it using the EKS cluster and then a bunch of CI/CD pipelines that you can use. And that's essentially is our work in progress pipeline, if you may say, that we are actually continuously developing and improving and getting it to a place. I think with that being said, I think it's you, Jen.
Thanks. I just wanted to close out with some of the lessons learned. So we do have a lot of machine learning models that are embedded in our small molecule drug discovery process. And our next steps will be really moving from those point solutions. I think what we have, and I think it was brought up in your lab in the loop presentation, where how do you bring this together to really be an AI native approach to drug discovery instead of it feeling like this handoff, so that you're going to do your design here, you're going to do your target validation here, then you're going to bring it over and you're going to do your ADME predictions over here. We do have a platform where we have all of our models embedded for all of our scientists to use, but there is a bit of just a workflow mind shift that has to happen when you start to think about having a true AI-infused end-to-end process. The other thing we are going to be doing in the coming months is evaluating some of the newer biological state-of-the-art foundational models, so working with evolutionary scale to look at ESM3, not only in the small molecule world, but also with proteins, antibody engineering, et cetera. So for lessons learned, start small, but aim big. And that is really speaking to ensuring that you understand what type of a model you need and what data you have. So you shouldn't always think that every use case requires the most complex, largest model to be deployed, because in some cases it isn't. It can be a very small local model that is very specifically trained on certain data sets that actually answers the question that your scientists have. So just ensure that your use case meets the models, or the model meets the needs of the use case, and don't always think that bigger is always better. Investing in culture, I think we've heard that a few times, both at the keynote as well as in today, where you have to take the stakeholders along with you. There is a big cultural component and a big change management curve that happens as we start to employ more machine learning. So ensuring that the scientists or any of your stakeholders really understand what this machine learning model is doing and how it actually makes their work more efficient or leads to higher probabilities of success. User experience is key, right? Embedding that into their day-to-day workflow. Do not make it command line outside in some other system, right? You don't want to be bringing up Linux. You want to be able to put it right into the software that they're typically using. So the user experience is really key. And last but not least is governance. And a lot of times we think of governance as putting the no in innovation. But governance can be really key. And that's to ensure that really what you're building is fair and that it is deployed in a correct way, that you really have transparency. Governance can be about making sure that you as a company are working on the right things and not just working on things. So governance is also something that's really important. And we do have a really nice framework at AbbVie to do our AI governance. So that is it. I would like to thank everybody for your attention. And I think at the end of the day, as I come back to really that North Star of patient-centric approach at AbbVie, all of this work that we're doing in the machine learning space and embedding artificial intelligence throughout our research and clinical and on-market products, it's really to make sure that what we're delivering are better medicines that are safer and getting those to patients faster. So thank you.